This video is about transcription, and transcription is the first step of protein synthesis, and this is when DNA becomes RNA. Protein synthesis is the process by which genetic code puts together proteins in the cell. DNA can't leave the nucleus, so the DNA has to be replicated in the nucleus, then the copy of the DNA can leave. So this is the DNA right here, and the DNA is read from the 3' end to the 5' end. The first thing that happens in transcription is initiation. The second messenger initiates the copying of the DNA after it sends the amplified message to the nucleus. The promoter and/or initiator comes down to the 3' end and latches on. The promoter latches on to the gene for the protein that it has to make. So here's the gene right here that it latched onto. The promoter latches on to the TATA box, and the TATA box is just a place in the DNA where there are a bunch of adenines and thymines, which begin the process of breaking the hydrogen bonds between the two nitrogen bases using the enzyme helicase. This section right here is now called the open complex. This section will code for the protein that is needed. This part is called elongation. After the hydrogen bonds are broken, the RNA polymerase, which is this pink thing right here, that is latched on to the TATA box, will read the code in a 3 to 5 direction. The pre-mRNA strand right here makes a copy of the DNA in a 5 to 3 direction. It makes an opposite copy of the nitrogen bases, making uracil for adenine, cytosine for guanine, and so on. A stop codon is a series of letters that indicate where the gene ends to stop the transcription. So right here would be the stop codon. And then the last thing that happens in transcription is termination and processing in eukaryotes. A 5' cap is put on the 5' end. So right here, there would be a 5' cap. And this is the front of the pre-mRNA strand, and it will leave the nucleus that way. It'll leave where the 5' cap is. And a poly A tail is put on the 3' end, which are just extra nitrogen bases that are put on the end to make a tail-like structure. The tail is to serve the purpose of protecting the code right here that is needed, because if parts of the tail break off, then the code that is needed right here will still be intact. Some of the pieces of the pre-mRNA strand codes for the protein that is needed, and some pieces don't. Important pieces are called exons, and unimportant pieces are called introns. So this section right here might be an exon. This section right here might be an intron. And then this section right here might be an exon. So the unimportant pieces are spliced out and eliminated. So all the introns are spliced out. And so then this pre-mRNA is now mRNA after the processing and termination is finished, and it will leave the nucleus that way.